Welcome. This is College Bio's 20th Annual Current Events Symposium. I am Mrs. Schoonmaker, and our presenters today are here in the audience, and they are current juniors and seniors in Ellenville Junior High School, and they are taking College Bio 2. They're also students at Ulster Community College, as they are dually enrolled there in the General Bio 106 program as part of our collegiate program. I'm so happy you've decided to join us today. We've been holding this event for 20 years now, and I think it's now kind of like a rite of passage for everybody who takes this class. We just have to get through. It's sort of a challenge, and here we are. It's today. Not only do we use this to meet the college's requirement for completing an independent research assignment, but we can use these projects to see where the most current science research ties in with the fundamental topics such as cell theory, genetics, homeostasis, and evolution that we've studied over the past 7 months. It's important to realize that having a firm grasp of the foundations of biology will allow us to understand where this current research, even now, and research in the future, can fit into society. In addition, public speaking is critical. And in this day and age with technology, I am well aware that it is much harder for our students to get up in front of groups and to give these kinds of presentations. So it's a great opportunity. You're going to have to do this in college. If you 2-year, 4-year, even if you're at a trade school, you're going to have to work collaboratively. You're going to have to work and present in front of other people. No better time than the present to give it a practice. Again, thanks so much for coming. What we ask is that you listen, that you're quiet and attentive. We absolutely encourage you to, if something you hear makes you want to ask a question, that you hold your question right until the end. At the end of presentations, we're going to open up the floor to questions. We will allow you to ask. What we will remind our presenters, make sure you repeat what you're hearing as the question so that we can get everything on camera, so we don't have a mic to pass around. So you'll ask, they'll repeat it, and then they'll try to give you sort of their best answer. I'll also be asking a few questions. Anybody who's here, certainly welcome to ask questions. The way I'm grading it, so in case you're curious what I'm doing over here on the side, is not only am I grading like content, which I've kind of proof checked all their content, it's really good stuff, but I'm also looking at eye contact and elocution. Can you understand what they're saying? Are they speaking in a way that's slow and understandable? So lots of different components to this particular presentation, and there's a time component. The time is from 4 to 6 minutes, so I've got my phone out. I will be running my stopwatch. The goal is no shorter than 4 minutes, no longer than 6 minutes, and you will be, their grade will be adjusted on either end. They lose a little bit of points if it's too short, they'll lose some points if it's too long. So a few of them have been down here practicing, so this is like any other skill. Public speaking and presenting is like any other skill. The more you practice, the more comfortable you will be and the more success we're going to find. Without any hesitation, I'm super excited to invite up our first presenter. Anthony Murdoch is going to talk to us a little bit about some current findings around fibromyalgia.
Hello, my name is Anthony Murdoch, and I'm going to talk about fibromyalgia and its current classifications. First, we can discuss what fibromyalgia is. Fibromyalgia is a chronic disorder which affects up to 4 million U.S. adults today, and its symptoms include hypersensitivities to hot and cold symptoms, like hot and cold reactions, hot baths, cold showers, and also more sensitivity to pain. Like you will feel pain in a more advanced level than the average person. It's very, very unknown whether what it is. It can be an inflammatory musculoskeletal disorder or an autoimmune disorder. We have no current cures or treatments that are very efficient for solving fibromyalgia and its pain. Like I said before, it has no current cures or treatments, but it can be prevented in some sort of way. You can have painkillers such as amitriptyline, which helps block pain receptors, which help have the reduced pain in itself. The very current at-home remedies include acupuncture, yoga, meditation, and some people exercise or sleep to help. Like I said before, it's either an inflammatory musculoskeletal disorder or an autoimmune disorder. They both have very different classifications which help each other. They are both incurable, but inflammatory musculoskeletal disorders are preventable because they include such things as arthritis and like osteoporosis and such. Autoimmune disorders are incurable. They are like when your own cells attack your own cells. Some inflammatory musculoskeletal disorders can be classified as autoimmune disorders such as rheumatoid arthritis because they attack your own cells. Autoimmune disorders are very this research I'm covering today is classifying fibromyalgia as an autoimmune disorder. They did this by infecting mice with serums from patients which have fibromyalgia and a control group of patients who do not have fibromyalgia. After injecting the mice with fibromyalgia, 24 to 48 hours later, they started showing symptoms of fibromyalgia, which include hypersensitivities, hot and cold, fatigue, and trouble sleeping. All the mice began within 24 to 48 hours, and they stopped showing symptoms after 1 week. They discovered the hypersensitivities to pain with the Rambo Stiletto paw grip strength test, which includes having a pressure plate, which is it measures how much pain perception they feel. Once they figured out that autoimmunoglobulin causes some hypersensitivity to hot and cold pain, they began to test and theorize of what could be used to help cure and stop fibromyalgia. They began doing this by increasing the dosage of the autoimmunoglobulin, which showed how much more perception of pain it has. So the more autoimmunoglobulin you have, the more pain you can perceive. So they began to theorize with mice, when they release the dosage, it has less pain. And also when they increase antibiotics or painkillers which block the perception of pain, they also decrease the amount of pain that they feel. On the top diagram, you can see that when the mice that are infected with fibromyalgia have decreased locomotor activity, which is evident of fatigue, which just means that they're not moving as much and they have more pain moving. And the bottom one is the hypersensitivities to pain, like their pain threshold and such. And that's all I have for now. Okay, what's your question?
Why did you pick this topic?
I picked this topic because my mother has fibromyalgia.
Okay. And did she give you some insight into what she deals with on a daily basis?
Yes, I got the at-home remedies from her. She has practiced acupuncture, massage, yoga, and meditation to help with fibromyalgia.
Okay. How about, so if you had to take this topic that you spoke of, which was very interesting, and link it to what we've learned in class, where would you put it in our school year? What chapter would you add this to?
I would put this with I feel like it could go within biochem because of how the receptors and how we learned how the plasma membranes and stuff and the receptors are on top with the increased pain and perception.
That's a great answer. Thank you.
So I was curious, when one of the symptoms you described was decreased movement, I'm imagining that's in response to the pain.
Yeah.
Could that be part of why things like massage and yoga help with it, because they involve movement, or at least massage involves increased circulation?
Yeah, that's why it helps. Massaging increases circulation, which helps reduce the amount of pain that you feel and the amount of receptors that are being used.
Thank you.
Yes, Ms. Deschamps?
So is this a disorder that somebody's born with, or does it develop? Like, are there environmental triggers?
You are born with it, but you don't feel as much symptoms until you become later in life.
If there's no more questions, thank you. We'll give you a round of applause.
Hello. For those of you who don't know me, my name is Caitlin Evans, and today my presentation is on the rule of flight and its correlation to evolution of modern-day birds and dinosaurs. The rule of flight was discovered by scientists from looking at hundreds of birds in different museums around the world. Seeing birds have similar traits, scientists found that all flying birds share a trend in their feathers. Having asymmetrical long primary feathers along the tips of their wings and having between 9 and 11 of these feathers. Primary feathers are the largest flight feathers on a bird's outer wings attached to the hand bones. Possessing these two traits is the rule to being able to fly. The scientist involved in finding this rule was a collaboration between Jin May O'Connor, who was a paleontologist, and Yoster Kite, who was a postdoctoral researcher. A paleontologist is a person who studies the record of life of Earth left as fossils. And the postdoctoral researcher is someone who is conducting research after the completion of their doctoral studies. These two scientists working together led to the rule of flight being discovered in birds. The rule of flight that was found, the scientists were able to figure out which dinosaurs were actually capable of flight, and the rule led to clues on how dinosaurs' ancestors of modern-day birds first evolved the ability of flight. Looking at multiple dinosaurs that had feathers, it was shown that not all were able to fly. Using the rule of flight, scientists looked at the number and shape of the primary feathers of fossils to determine whether it could fly or not, and to see if its ancestors had the ability of flight as well. Two examples of dinosaurs with feathers would be the Penaraptorians and the Velociraptors. Even though both of these dinosaurs had feathers, only the Penaraptorians were able to fly. It was shown that both of these dinosaurs had developed feathers before the ability to fly, which led scientists to assume that the original purpose of feathers was not for flight, but for insulation or to attract mates. Comparing Penaraptorians fossils to traits of the rules of flight showed that they were able to fly and have similar traits to modern-day flying birds. While Velociraptor fossils can be more closely compared to flightless birds, such as penguins and emus, who have symmetrical primary feathers that have a number of feathers outside of the 9 through 11 seen in the rule of flight. In conclusion of this presentation, the learning of this rule of flight can better the understanding of evolution from dinosaurs to birds, as well as the evolution of the ability of flight. Thank you for listening. I will now answer any questions you have to the best of my ability.
I have questions. Why do you think that the dinosaurs and then subsequently the birds show flight at any point? Why is flight so important? Why do you think? What do you think?
That's a good question. I think flight is so important because, like back in the dinosaur age, there was a lot of predators around. So being able to take to the sky would be a very useful research to escape.
And then my same question that I'm going to ask all of you. Where would we put this in our class? If this is a great topic, we've discussed some of this, where would you have added it into our coursework? In what, think what chapter?
Well, the chapter we're working in right now within sponges and the animals, since this is evolution, it correlates a lot to that.
Okay. Thank you.
Yes.
In any of your research, did you find out why the asymmetry in the feathers helped them fly?
Unfortunately, the research didn't go over that, no. Any other questions?
Yeah, come on. You want more?
Thank you.
I'm Gina Stewart, and this is an IVF breakthrough. Brief overview, what is IVF? IVF, or in vitro fertilization, is a medical procedure where a human egg is fertilized by a sperm outside the body. The question this research was looking for an answer to is, why do fertilized human eggs often falter early on? Compared to other species, human reproduction is dramatically less efficient. Many scientists from genome medicine work together to figure out why exactly this is. The experiment. In the experiment, scientists collaborated to analyze almost 1,000 embryos in vitro fertilization, or IVF. More specifically, they focused on the effects of aneuploidy for 909 embryos and performed trophectoderm biopsies, which help look for specific gene defects. The results. The results of this experiment showed that mitotic and meiotic aneuploidy, which aneuploidy is having an abnormal number of chromosomes, which means you could be missing or gaining some. This was common in 94% of arrested embryos, and when postzygotic cells were transitioning from the cleavage to the blastocyst stage, aneuploidy is most likely to occur. And this is just a diagram of what aneuploidy would look like. Meiotic aneuploidy unevenly impacted chromosomes 15, 16, 19, 21, and 22. However, mitotic aneuploidies dominate amongst all chromosomes. It was also found that meiotic aneuploidy was more common with greater maternal age. However, mitotic aneuploidy had no relation to maternal age. How does this benefit society? This study has clarified that aneuploidy is the main reason for high early embryonic immortality rates. Knowing that this is the main cause, and the fact that the transition from the cleavage to the blastocyst stage is when this most often occurs, can help improve the IVF experiences for many couples, and IVF providers will be able to focus on the transition and notice if any abnormal cell division is happening. Further questions? Now that the root of the embryo loss is found, the next question is how or can this even be prevented? Is there any way to control it? Some scientists have been looking into the environment in which they fertilize the egg, and if that could be a factor of this abnormal cell division. Personal feedback. I find that this is a very amazing discovery. They have found the root cause of early embryo immortality and when that occurs. This research can help innovate the IVF process, which will then allow couples struggling to conceive have a better chance of finding that child of their own.
So, one thing, you talked about both mitotic aneuploidies and meiotic aneuploidies. Can they ever occur at the same time? So you talked about them separately, but
No, they do not occur.
they don't occur at the same time. Can you imagine the amount of
Yeah.
damage that would happen? Was that all happening? What is the difference? When you talk, like, if you're looking at a mitotic aneuploidy, so you're adding or subtracting numbers of chromosomes. When it's mitotic versus meiotic, do you know what the difference is?
Well, meiotic would have to do with like the mother, so the mother would have to have that aneuploidy happen to her and like pass on to the daughter cell, or the father, either one. But mitotic would happen with the zygote, and their like genes would like.
So it's a time.
Yeah.
And then finally, where would you include this in our course? If we're teaching college bio, where does this fit in?
Probably cell division.
Yeah.
Yeah.
So we did cell division a couple different times, and we just did this. Would you do a first semester or second semester? do you think?
Probably first.
First semester, third semester. Thank you.
Should I ask a physics question?
No.
So you mentioned that this research is into like the IVF success rates, and oftentimes couples that are looking to go this route are in fact older couples.
Yeah.
Like, was there anything in your readings about maybe people looking to freeze some of their gametes earlier in life in case this comes up, or?
No, they did not talk about that in research. Any other questions? Thank you.
Alright, I'm Kevin Santiago, and I'll be talking about a new method of detecting pathogens by melting DNA. This new method doesn't really melt your DNA, it just unwinds it by a process called denaturation, which is the process by which DNA is heated to the point where the bonds within it break, causing it to unwind into two separate strands. Based on the amino sequence of the DNA, it'll unwind at a different temperature. And knowing this, the researchers developed a machine learning algorithm that has access to a database of pre-existing temperatures and was able to discover or detect and diagnose different pathogens based on that temperature. What we currently use is a method called blood cultures, which you take a blood sample and place it into either an anaerobic jar or an aerobic jar, and it is left to grow the bacteria, fungus, or yeast within the sample. And then it is sent off to a lab where then the bacteria within is identified. A gram stain may also be used to determine the bacteria. These can take from 15 hours to several days to get results back. The current method the new method, digital DNA melting analysis, is far more faster and accurate. 1 out of 3 blood cultures will show a false positive, while the new method showed 0 false positives. It is also far more faster, with it being able to show results in under 6 hours. Why this is important to us. So, sepsis, a infection caused by blood-borne pathogens, which could be either bacteria or viral, is the leading cause of death in U.S. hospitals, and is responsible for 20% of all deaths globally, which is about 1 out of every 5 deaths. Roughly every hour, sepsis is untreated or mistreated, which happens often due to false positives. The mortality rate can increase by 4%, but if it's treated properly with antibiotics, it can decrease by 7.6%. 1 out of 3 patients who die in a hospital will die as a result of sepsis-related complications. Here's some more information on sepsis. There's about 50 million cases a year globally, and at least 11 million deaths from sepsis. Most of these, like I think it was around 42%, are in children under 5 years old. If this were implemented even just in the United States, it could save thousands of lives. I think there was 200,000 deaths last year due to sepsis. That was all. Is there any questions? Yes.
Do you know if our local hospitals have access to this new?
I don't believe they do have access to it because it is pretty new. This is like only a couple months old.
Oh, wow.
I think the article came out in June of last year, on the 15th, I think.
Did you watch the movie on Netflix about the guy who had sepsis? He was in, I think, Britain somewhere?
I have not, but I might have to check that out.
Well, be prepared. It makes some sense.
Yes.
So this is a diagnostic procedure, correct? So what
Yeah.
does that mean? If you're diagnosing it, it means what?
You're determining whether or not they have sepsis.
Right, so we're using it as an identification tool. What if what if somebody's sick with say they've been infected by a pathogen that we've never seen? Does this procedure still have value?
Yes, so it'll show up. If they won't know what it is, they'll have to either pull that sample aside and look at it like specifically, or if it's like a mutation or whatever, then they'll have to identify that. It'll still show up, it just won't be like identified. It'll just be unknown.
But if you're not sure what's making somebody sick, and now we've got this unknown pathogen that we can't identify because it doesn't fit our algorithm, you can at least...
Yeah, I mean, the same thing happens with blood cultures because it amplifies any DNA present. So it's the same thing, they just wouldn't know what it is.
Okay.
They would have to look at it deeper.
Okay, and then just two more questions. One, so if this, what did I want to ask you? Well, why did you include aerobic and anaerobic samplings? Why would that be important,
So
biologically speaking?
for blood cultures, it helps narrow it down because some bacteria thrive in either oxygen-rich or oxygen-lacking environments, which anaerobic or aerobic have either. So there's the two types.
And then finally, where would we put this in our course? If you had to share this information, we're going to include it in our class. Where would you?
When we talked about viruses and bacteria, there's a pathogen, bloodborne pathogen can be either viruses or bacteria. It could be either.
So we can put it right in there. Are there any drawbacks to this? I know you talked about a lot of benefits. It's faster, it's more accurate, more reliable. Were there any drawbacks?
So I tried to find like a price because the way method by which they use it is like a tiny, it looks like they ever use a microscope, like the glass slides, it looks like that, and you put the blood on one side. That seems pretty hard to make, so probably expensive, but there is no information on it yet. That's all, thank you.
Wait, no.
My bad. Yes.
So you said it's still in the introduction phase, but how many different pathogens, were they testing? Can they identify at this point?
So the research article that I used only tested on sepsis patients. They didn't use any, no, they used only sepsis. But I'm sure any, yes.
In your research, was there anything about what would happen if there was a cooler temperature use?
So can you say that louder, please?
In your research, was there anything that said what would happen to the sepsis if a cooler temperature was used?
For colder temperatures, nothing about that. If DNA is denatured and then cooled down, it just rewinds itself. Any other questions?
Right. Listen, thank you so much for coming today. It's really exciting.
Hello, everyone. My name is Madison Pauly. Sorry about that. And today I will be discussing the tuberculosis vaccine and how it may be possibly used as a new form of cancer treatment. Now, liver cancer is the third leading cause of cancer deaths and worldwide and one of the most hard to treat cancers. Today, cancer treatments like chemotherapy and radiology have low success rates and many patients experience damaging side effects. This has caused researchers at UC Davis to try and find a new way to create a simple yet effective treatment to decrease liver tumor mass in cancer patients. Researchers believe that by giving patients a dose of the BCG vaccine, a vaccine commonly used to treat tuberculosis, it would stimulate an immune response to the cancerous tumor. They first tested the vaccine on mice with liver cancer, and to their surprise, their hypothesis was correct. The tumor mass in the mice had decreased. It was discovered that the BCG treatment results reduced inflammation in the body and prompted the movement of T cells and white blood cells to the tumor. The study also found that the vaccine is not a sex-dependent treatment, and both male and females responded the same. This type of immunotherapy offers an alternative to other futile cancer treatments. Not only has BCG been safely used around the world for many years, it is also believed to be far more superior than any cancer treatments. In animals, BCG has produced more successful outcomes than any other standard immunotherapy, and the treatment itself only requires a single injection, making it a much more simplified treatment plan. While this discovery is highly significant, there are still some questions that need to be addressed. Researchers are still unsure of the efficiency of the vaccine over time, and they also are unsure of whether the treatment will have an effect on the gut microbiome. There also needs to be an exploration on whether multiple doses or boosting the dosage amount would be more effective at treating patients. This upcoming form of cancer treatment shows promising results, and I believe it has the possibility to help many cancer patients around the world. Patients who have had complications with previous immunotherapies will finally be able to experience a simple and effective form of treatment. I hope this new form of treatment will be available for use soon. And that's all I have for you guys today. Thank you.
Okay, so first question, how does this fit in with what we've been talking about? Explain.
I feel like this fit, like what topic or like just how?
Yeah.
I feel like, personally, I feel like it would fit into probably viruses and bacteria because it does talk about like the signaling of T cells and white blood cells and, you know, how they like attack those pathogens. So I guess, yeah, I'd fit it probably into somewhere there since we discussed that a little bit.
Okay. And then in terms of like it's an effective treatment for cancer. Is it? This is a new vaccine, or this is a standard TB vaccine?
This is a standard TB vaccine. They've been using this for many years prior to this. I think they're just realizing that this also might be used as a form of cancer treatment as well. I think they're just kind of researching that. But yes, this has been used like multiple years safely around the world for
Okay, so you don't have to deal with that. You're just dealing. Does it have side effects?
As of right now, they didn't really touch much on the side effects. They mostly just were touching on how it's very simple. I think one of the things is that it doesn't come with the side effects like the other type of cancer treatments like chemo, radiology, stuff like that. So I think that's another thing why they were pushing so hard for this, because it was just a lot more beneficial for the patient. Yes.
Do you know how they obtain rats with liver cancer?
I'm sorry, can you say that again?
Do you know how they obtain the rats with liver cancer?
I'm sorry, I just can't understand.
You know how they attack the rats with liver cancer?
Oh, how they obtain them? Honestly, I have no idea how they get them. They didn't really tell us where they got them. I'm assuming it's just probably from like lab. Yes.
So I know the TB vaccine is more targeted for, like a lung disease.
Mm-hmm.
In the research, did you see how they found like the relation, that this could be a treatment for?
I think the relation was that they wanted to use this vaccine because it is derived from a bacteria. And I think what they were thinking was that the bacteria would kind of signal the T cells and stuff like that to attack the tumor mass. So I'm not sure, they didn't really talk much about it having to do with like lung treatment. They actually talk more about it with bladder cancer. I guess they've been trying it with bladder cancer as well. Something with that as it also being beneficial for bladder cancer, but not really much about lung treatments.
Anybody else? Oh, you got another question for that?
Go ahead.
Has it been like tested for how well it would work as a long-term treatment yet?
See, that was actually one of the questions they had, was how efficient it will be over time, and they still do need to explore more into it on whether or not it will be efficient over time. So, all right, thank you.
Good afternoon. First and foremost, I want to thank everybody for coming out. I greatly appreciate it. Ms. Schoolmaker, I want to thank you for giving us this opportunity to share our knowledge and our learnings with these underclassmen and adults. So my topic is on cryotherapy, and what it basically is it's a medical treatment of cold temperatures used to treat like sports injuries or a variety of medical conditions, but it's good for swelling, pain relief. But before I get into the science behind the cryotherapy, I want to talk about what influenced me to pick this topic. For those who don't know, I was on the basketball team for 3 years, and this past season was my senior season, and I had around mid-season, I had a ankle sprain, which was probably the worst one I ever had. And as an athlete, your initial thought when you get hurt is, can I still play? And if not, when can I play? But my initial thoughts were to keep playing no matter what. And the result of that was very painful. I went home, could not walk. And it caused me to miss the next 2 practices and the next very big game of the season. And it made me try to find out how I could get back on the court quicker. And mainly what I did was ice first, ice bucket, and then after, hot water, Epsom salt, stuff like that. But what I found useful was essential oils, like arnica. It's like a really good oil to soothe the muscles and get flexibility back in the muscle. And I found that really helped me the most. Other uses for cryotherapy, as we know, it's good for swelling and inflammation, but it also boosts the immune system. It produces white blood cells, which can help fight infections and diseases and stuff like that. It's also good to treat skin conditions like eczema, psoriasis, and acne. And it's also good in cancers like liver cancer, early stages of prostate cancer, and lung cancer and skin cancers. Older studies show, as we already know, inflammation, and it helps. It was said to speed up the healing process by producing, it's supposed to stimulate blood flow through the injury, which allows it to heal faster. But newer studies show that it's the complete opposite. But our typical treatments are cold therapy, elevation, compression, stuff like that. Compression to take the pressure off injuries, sports injuries. But as we see here, new studies concerns are frostbite. Too much exposure to cold temperatures can cause ice crystals to form in cells and injuries. But it also delays the healing process because the cold temperature acts as a vasoconstrictor, which is basically how the blood flows through, and the cold temperatures affect that blood flow through the injury, which delays the healing process. Throwbacks, I would say, are studies have shown that there haven't been significant benefits of cryotherapy, even though it is really popular, especially in sports, and professional athletes use it the most. There have not been significant benefits, but other studies have shown mixed results of it. Alternatives, like I said, essential oils such as arnica, which is what I use, it is good for soothing muscle aches, and it is good for reducing inflammation. Pain relief creams, which is similar, it helps with function and allowing flexibility through the muscle, and it's supposed to speed up recovery times. And then your typical drug therapies like aspirin or Advil. And that is it. Any questions?
Questions?
Yes.
One, so you said that you could use cryotherapy for treating cancer. Did you read how that would work?
Yeah, so basically what I read, I tried looking deeper into it, but it was more of the same result. What they do with cryotherapy in cancer is they just freeze the cancer cells until they're destroyed. And skin cancers, they just put liquid nitrogen on the area of cancer.
So it's kind of like the same as a treatment for a wart.
Yeah, basically.
Kill the cells and get them out.
Yeah, they talk about that in research too, with warts and how it's used for any skin conditions.
Okay. And then where would you put this? If you had to take all this, you say, all right, Mrs. Connect, we're going to add it to our class.
Biochemistry. I would say because it talks about, it's more like protein synthesis, and muscles are proteins, so put it around here. That's the best I got. Any other questions? Yes.
So for cryotherapy, would you need a sprain or a really bad muscle cramp?
It's mainly for muscle injuries or joint pains or any type of ligament injuries. That's really what it's used for. But it has other health benefits. It's said to improve breathing and any type of lung or whatever. It's decent.
Did you read anything about them addressing the contradictions of, cold being a vasoconstrictor but also saying that like increased blood flow will help speed up the healing process?
I think that's where the heat comes in, because I read that the cold is just for pain and swelling, and then once you put the heat in, it's what relaxes the muscle and then increases the blood flow back into the injury, which allows it to heal. But that's as far as I got.
So that's, I think like a lot of people mix like a cold plunge with like a sauna type thing.
Yeah.
Okay.
Any other questions?
I just comment because I read a little bit about this, and there's sort of like a difference between injury treatment, which has the hot and cold like alternations, versus pain, say like body pains, like the daily use body pains, like that's what those big like full immersion ice baths are for, like maintenance rather than treatment. And then you do the cuffs and things like those compression ice cuffs where you alternate back and forth with heat if you have like a specific injury you're really trying to deal with. So it kind of does a little of everything.
Yeah. Any other questions?
Hello, my name is Jeremy Lopez, and I will be talking about meat grown from cells, that being lab-grown meat. Lab-grown meat is grown from cells, whether that's from the tissue of a living animal, such as a muscle, a fertilized egg, or a cell bank. This process tends to take between 2 to 8 weeks, and it's done by taking the cell and bathing it in a nutrient-rich broth, in which it is placed in a bioreactor for the cells to multiply. This has become the next alternative to livestock, that being because livestock contributes between 10 to 20% of all greenhouse gas emissions from cows. Cows, just by living, tend to produce anywhere between 154 to 264 pounds of methane gas per year. Currently, there are limitations for this new innovation, that being only 2 nations are actually allowing for this, that being Singapore and the United States. There's also cost problems that come in hand, that being because the production of just 1 pound tends to cost between $17 and $23. They would need to drop the price by roughly $10 or to $10 to make it commercially available so that people like me and you could go out to the store and buy it straight off the shelves. Despite this not being widely available, it does have potential to replacing livestock and not needing to kill animals and damaging the environment just to feed people. This is an innovative way to change the diet and not cause as much harm as we do to the environment.
Right. Yep. Would you eat that? Would you eat it?
I would absolutely eat it.
Would you? So a meat that is grown. Okay, that's
No problem.
Where would we put this in our class?
I would put this in cell division, and right now we're actually learning about animals, so I'd also put it in there. Do you have any questions? Yes.
What type of cell is it grown from?
All right, I was asked what type of cell was it grown from. This could be from the cell taken from the tissue of an animal, like the muscle, the fertilized egg, or a cell bank in which cells are stored for either research purposes or just to have it stored. Yes, Ishan?
Do you think, say like a buy-in from a large company like a McDonald's would cause the price to drop down and make this more widespread and acceptable?
All right, Deshaun asked me if companies such as McDonald's, if they were to use this, if it would drop the price. It could drop the price, as some of these companies, roughly 150 of them, they plan to get it within restaurants within 5 years, that being because it's cheaper, or to get it within the shops within 7 to 10 years. Yes?
What cells have been like grown to intestinal? Like what cells have they grown?
You're asking from which animals, right?
Yeah, from which animals, yeah.
It's most easy for ground beef, and chicken has been done a lot. Any frozen product like that.
Have there been any like taste test comparisons that you've read about?
There has not been much taste test comparisons, that's just being because a lot of people don't like the idea of it. But that's also one of the hurdles they have to go against because they have to make it either taste the same or just have it taste even better, which is hard to go around.
Does it, like, when it grows, what does it look like? Like, is it just a mound of cells?
That's a great question. This is actually it's not just going to be like a mound because they use a scaffolding type of thing where they have a collagen or use a vegetable-based source so the cells can grow around. I'm sorry, what was that?
You know how long it takes?
It actually takes anywhere between 2 to 8 weeks typically, some even taking several days, but that's the quickest it can be produced.
Would you guys eat it? Meat grown in a lab? No. Mr. Kroger says no. I love
All right,
this.
any more
Jeremy,
questions?
thank you so much.
Hi, I'm Amelia, and I chose to do research on new technology to see how other species see. Like, in this picture, the butterflies are how a bird sees them. Okay. This was just recently found on January 23rd of this year. In this research, we found that video recording and analysis technique help scientists be able to understand different types of species and how they view the world, such as birds, butterflies, honeybees, mice, and more. This inexpensive technique is used by a combination of specialized cameras and a 3D printed enclosure with a beam splitter. One camera is sensitive to UV light while the other is sensitive to human visible light. They align the footage to see what each species sees. The top bulb is UV light and the bottom bulb is visible light, and I'm pretty sure that's the camera. Why does this benefit us? We can see life through other species' eyes. It gives humans a better understanding of ecology, interactions with the environment, and how it can help light pollution with insects. It helps improve technology with video recording, and it can help humans who have disorders make their lives better, and altogether improve the understanding of the vision. New questions we can ask are, what evolutionary pressures led to this development and how they see different? Why do animals use their unique types of vision? How can this new video recording be used for other studies, and how can these insights be used to build better life for species? Personal feedback was I thought it was cool because this new invention, I thought this invention was cool because I've always wondered how my dogs see, and now it might be able to know, like, really, I see like filters and stuff, but you don't know if that's accurate. And I can know what will attract animals like birds, and I hope this research will bring more thought to living species and how they see.
How does this fit into our curriculum? What science did you learn from me that you helped you with this?
Well, probably what we're learning now, like animals, but like ecology, because it's animals and what we see.
Why is it important for, like, so you said, well, this is how the bird sees the butterfly. Why is that important to know? What does that help us to do, David?
Like, they have like different wavelengths, and they see like different colors. Like, I saw a picture with like a flower, and like the butterfly is just like I think it was where the pollen is or something. It's like like a lighter color than the rest. So I feel like that's important because we can see like what brings them more and what goes out more to them. Any other questions? Mr. Duchamp?
just first, I've seen similar things with this where some birds, to our eyes, both the males and females look the same, but when you put them under ultraviolet light, there's like stark differences within their wavelength that they can see. So, you know, like, so those birds, they look very different. Did you find anything with your, like, the flowers or like the butterflies? Like, do the butterflies, are they able, like, to look different to themselves, I guess, is one way?
I saw something about like peacocks, how they see different, like their feathers are way more colorful than we see them, but that's the only thing I saw about how other birds will see themselves.
My guess is that would have a lot to do with like reproduction. Like if you can, the different colorations is one of those external signs of here I'm looking for a mate. Did it show? You saw just the butterflies? You said you were interested about the dogs. Did you find anything out about your dog?
No, I tried to look it up, but it's like so new. They don't have any pictures of like anything there. So like, I don't know. So it's like, there was, that is how, I'm pretty sure it was how a butterfly sees a caterpillar. But yeah, that was it.
Great. Thank you.
Hi, my name is Kaylee Kane. I chose to touch upon is Crohn's disease. Okay. What is Crohn's disease? Crohn's disease is a type of inflammatory bowel disease that causes swelling of the tissues in your digestive tract, which can lead to many symptoms. Some of the symptoms include fatigue, abdominal pain, reduced appetite and weight loss, anemia, and kidney stones. The main question is, how does someone get Crohn's disease? Although doctors have not found a direct cause of people diagnosed with Crohn's, there are many aggravations that lead to the diagnosis. The two main aggravating factors being your immune system and heredity. Your immune system is fighting off bacteria or other viruses. It attacks the cells in your digestive tract, which is mistakenly attacking healthy tissues, which can cause a Crohn's disease. Heredity is very common when someone gets diagnosed with Crohn's that a family member also has it. However, you can get Crohn's disease with no genetic trait at all. Ongoing research to find a cure. One of the hardest things for people with Crohn's is you have to have it for the rest of your life and there's still no cure to treat the patients. Meaning new research and medications all year round are being tried. One of the most recent ones being JAK inhibitors. Janus kinase inhibitors is a type of medication that block inflammatory pathways that drive diseases such as Crohn's disease. JAKs are kinase proteins that are linked to cytokine receptors, sending a message to other pathways. These pathways can include pivotal, intestinal homeostasis, and inflammation. Without the ability to control JAK inhibitors within the pathway, leads to inflammation diseases such as Crohn's, which is why, with the help of controlling JAK, it can help some people, but if it's not controlled, it will not help other people. Okay, evaluations. The JK research has shown that it has positively helped some patients with Crohn's disease by putting 49.5% of people in remission compared to the 29.1% in the placebo group. However, like I said before, there are risks that come along with taking this medication. Some risks include infections, mortality, and cancer. People should also take other factors into consideration before taking this medication. People who are under the age of 65, smokers, or people who have a smoking history cannot take this medication because it will not help them and will not work for them. How is this research beneficial to society? Conducting this research has allowed some, but not all patients with Crohn's to feel some sort of relief. Like I said before, there's no cure to treat this disease at all. People who struggle with it will have it for the rest of their lives. I may sound a little bit biased, but my father has Crohn's disease, and when I spoke to him about it to get some insight, he has been on 4 different pill medications. He is currently taking infusions, which is from every 6 to 8 weeks, and he has to go for 3 hours, which completely shuts your immune system down. So it's very easy for you to catch other diseases and colds and flus and other things like that. New questions that the research has created, just like anything else, doctors are still wondering if it will continue to work, because an ongoing issue with Crohn's is that initially patients will have a positive reaction, but then maybe down the line it will not work anymore. That's why they're constantly trying new medications all the time, and doctors are still researching new things yearly, monthly, weekly. Once again, the question is why? There's really no answer. And that's it. I would put it in biochemistry, because in biochemistry we discuss the changing in your living cells, and in Crohn's disease it involves your white blood cells, your red blood cells, fighting diseases, things changing in your cells and everything.
And then the other thing I was curious is, so this is a new treatment. They're looking at it, I would assume, just in isolation. Do you think it would be, can you work in conjunction with other treatments?
Yeah. I know my dad, he takes multiple different treatments at once. So they all work in different ways. Like I said, it messes up your digestion. You can't digest them sometimes, or sometimes you can throw up what you ate a half hour ago, or it can affect you a week later. So they all go together. It affects probably like your whole body. Any other questions?
Is there a test for Crohn's disease? Like, what's the test?
There really is no test for it or not. There's multiple tests that leads you to the diagnosis. You go through many tests in general. I know when I spoke to my father, he said for a while doctors don't believe patients with Crohn's because, like I said, there's nothing that can track you down to getting it. There's a lot of things, and a lot of time doctors don't believe patients with Crohn's that they have it. They believe they're seeking something else, or they're lying, or because, like I said, it's an invisible disease. You can't see it. There's nothing that can cause it. It's just a bunch of tests.
Is it an autoimmune disease?
I think so.
Once you're diagnosed with Crohn's disease, are there dietary restrictions, things that people say you should avoid?
There's a lot of dietary restrictions. I know a lot of times you can't have a lot of red meat. There's a lot of other things like lettuce that can mix up your digestive. There's a lot. You can't eat a lot. I know he said now he hates food.
What would someone do to cause their immune system to get Crohn's disease? Like, what would someone end up doing physically?
To get Crohn's disease,
Yeah.
like what causes it?
Yeah.
Like I said before, you can't put one thing to it that causes it. Doctors have said that, like I said before, people who have a history of smoking, or if it is hereditary, but you cannot have the genetic trait, and you can still have it. There's no cause. That's why it's very, very difficult to deal with.
This just kind of reminded me of the fibromyalgia talk from yesterday, just how it's an autoimmune disease that we don't know a lot about yet. But do they share any similarities from what you heard from yesterday's presentation?
I think just like you basically just said, with not it being invisible, and a lot of times doctors not seeing it, and there's no cure for it. Right now, people that have both of those diseases just have to live with it. So I would say, yeah, they're very similar in certain ways.
College Bio, you did it, you made it. I know you guys were very stressed about it, but they were great. Audience, thank you so much. I'm glad you guys had so many great questions today. It was wonderful. You were a great audience. Thank you, all the teachers. Thanks for coming and seeing and just helping them to realize that this is not the end of the world. It was a really great experience. And I thought every presentation was really insightful, was interesting, and some of you just did some stuff that I think you didn't believe you could do, and yet you got up here and you did it. So I'm really proud of you. Thank you so much.